So, life cycle of Phytophthora. Phytophthora infestans is said to be a re-emerging pathogen as it resurges and causes recurrent infection to the same field and to the nearby fields as well as to every corner of the world. So, it is known as the re-emerging pathogen. This Phytophthora infestans causing late blight of potato was the cause of Irish famine when people had faced acute food crisis because the potato crop was totally ruined in 1845 and the people did not have enough food reserve and so they had to suffer severely. So let's see how it affected India and let's see how it affected West Bengal too. So here we see how the farmers of Bengal were left to the dismay and how they were totally shattered when the when the potato crop was destroyed by the Phytophthora infestans infection causing late blight in Bengal, and the entire crop was destroyed in 2014, and because of the huge financial loss that they had suffered in 2014, many of them were compelled to commit suicide because the government was reluctant to give any support for the or to compensate the loss they had suffered. So here we see the symptoms that are visible on the foliage as well as potato tuber when the disease affects the crop. Initially, the symptoms are seen on the leaf. Here we see there are some black delineated lesions that are necrotic in nature, and they are initially brownish and later on when they mature they become purplish or blackish in color. Then these the lesions they start from the midrib and they progress towards the margin and then subsequently to the tips of the leaf. And here we see the symptoms affecting the potato tuber. Here there are some brown markings on the potato tuber, and as the potato tuber they mature, these lesions they become water soaked purple and they emit some pungent odor, and when they are kept in cold storage, these affected tubers or the infected tubers they affect the other tubers that are kept along with the infected tuber in the same gunny bag. So now let's quickly see the systematic position of Phytophthora infestans. Phytophthora infestans, as if we look into the classification as done by G.C. Ainsworth in 1963, he had placed Phytophthora under Eumycota because it is a true fungi and it has a true cell wall and an organized structure, and the assimilative phase is cellular and hyphal and mycelial in nature. And in the next stage, it belongs to the subdivision Mastigomycotina, and the class is Oomycetes because they have been classified under Mastigomycotina subdivision because they have got some motile cells. Mastigomycotina differs from the other subdivisions in the presence of motile cells, and lastly, the class is oomycetes because they have oospore as their sexual spore, and they also have got zoospores as their asexual spores that are anteriorly flagellate, and these anteriorly flagellate zoospores are biflagellate, one of the flagella being tinsel and the other being of whiplash type, and they belong to the order Perinosporalis, family Perinosporaceae, genus Phytophthora, and the most infective and the most deadly pathogen, or the deadly species or strain of Phytophthora is Phytophthora infestans. As we by this time we already know, this is the causal organism of the deadly disease of late blight of potato. So now mm, let's see the life cycle of Phytophthora, how it thrives in the soil and how it encourages the disease spread and causes the disease cycle to move, and how it spreads from one field to another, and it asserts a epidemic state when it affects a particular field in the locality. So Phytophthora, since it affects crops, potato crops year after year, we need to know from where does the inoculum come from. It is the oospores that survive in the soil as dormant spores. These oospores, they have interior core, or they have a core made up of endospore, and the outer spore or the outer wall is known as the exospore. The exospore is relatively thin walled compared to the endospore, and but still this is a very tough structure, resistant to water and high temperature or low temperature, that is temperature fluctuations, as well as water stress in soil. These oospores, when they get favorable conditions, they overcome dormancy, and then they germinate to form the the sporangium or sporangiophore, and the tips of these sporangiophores, they bear the sporangium. This sporangium, depending upon the atmospheric conditions, they may undergo direct germination or indirect germination. If the conditions are favorable, that is, if the temperatures are below 15 degree centigrade and the humidity is quite high, that is, it ranges between, it is greater than 90%, and if it is between 90 to 100%, then these then direct germination of the sporangium occurs. The sporangium, in that case, the cytoplasm of the sporangium gets organized into conidium, and these conidium, they get released from the sporangium and subsequently form germ tubes, and these germ tubes enable them to lodge on the surface of the host, and when they lodge on the surface of the host, they either invade the internal tissues by forming apressoria or peg, and then they multiply and ramify within the host tissue, and in the intercellular spaces they form sporangiophore, and new sporangia are produced. So, this is the direct germination. But when atmospheric conditions are not that favorable, in that situation, that is, if the temperatures are between 12 to 15 degrees centigrade and the the humidity is much less, in that case the sporangium undergoes indirect germination. That is, the sporangium gets organized into some compartments as we see here, and these compartments are the future or the zoospores. The zoospores are formed by the encysting of cytoplasm around a nucleus, and they get organized or enclosed within a membrane or the cell membrane. And these zoospores, on attaining maturity, they are released from the vesicle of the sporangium, and on being released they produce, or they show their the two flagella, and they keep swimming in water, or they float in water, and until they reach a suitable host, when they withdraw their flagella and then produce germ tubes. And these germ tubes, like in case of direct germination, they help to invade the host tissue with the formation of appresorium, and subsequently by the peg, and these peg help to ramify within the host tissue. So these are the two types of germination. In the next so when these hosts they get affected, uh, when they get affected, zoospores are released from the either from the through the stoma or through the leaf surface. They are released, or they through the vascular system they migrate to the soil, and there they infect new tubers. These tubers are then the source of inoculum to the other plants, and through the soil water they migrate to other new tubers, and these tubers fresh mycelia produce sporangium. They produce